In this video, let's talk about how to determine protein activity.
Is the protein expressed in the supernatant necessarily active?
The activity of proteins depends on various factors, including whether the expression process is correct, whether the protein is sensitive to temperature and other conditions, and the influence of various experimental conditions during the later purification process. If a protein folds incorrectly during expression, or is affected by unfavorable conditions such as temperature and pH, even if it is expressed in the supernatant, it may not have activity. Therefore, protein activity cannot be judged solely based on supernatant expression.
Are all proteins expressed by inclusion bodies inactive?
Although proteins in inclusion bodies are usually non-folded aggregates and do not have biological activity, proteins in inclusion bodies may recover their correct folding conformation and biological activity after undergoing denaturation and refolding processes. This process requires precise operation and control, including steps such as collection, washing, dissolution, and refolding of inclusion bodies. By optimizing the conditions of these steps, the refolding efficiency and activity recovery rate of inclusion body proteins can be improved. However, not all inclusion body proteins can be successfully refolded and regain activity. This depends on the properties of the protein itself, the selection of refolding conditions, and the precision of experimental procedures. Therefore, it cannot be generalized that proteins expressed in inclusion bodies have no activity. In summary, the activity status of both supernatant expressed proteins and inclusion body expressed proteins is not absolute. It is necessary to verify and optimize through experiments to determine the activity state of the protein and improve its purity. This is the end of this video. Follow Cheng Yao Biology's video account to bring you more interesting and informative life science knowledge. We look forward to seeing you next time.